I'd like to welcome our speaker, Dr. David Giblin from the University of Washington Herbarium at the Burke Museum. David has been the collections manager at the University of Washington Herbarium at the Burke Natural History and Culture since 2002. His research focuses on the distribution and diversity of the Pacific Northwest vascular plant flora, and he has conducted extensive fieldwork in the region in support of that work. He is the lead editor and co-author for the one-volume Flora of the Pacific Northwest and a co-author of several wildflower guides and plant identification apps in the region. He manages both the Plants of Washington Image Gallery and the Washington Flora Checklist, and he co-manages the Consortium for Pacific Northwest Herbaria online database. In addition to his duties in the herbarium, David serves as a board member and taxon editor for the Flora of North America project. It's my pleasure to welcome him this afternoon for our webinar. All of you as participants have some tools and resources available to you, including a chat and a Q&A. We are recording this webinar today, and your names will be populated in the Q&A and in the chat should you choose to use it. We will have a 15-minute time period at the end of this presentation for your questions and comments. Thank you all for being here, and please welcome David Giblin.
Thank you, Denise, and welcome everybody. I'm on video for the moment, just to say hello, wave, and then I'm going to turn over my screen here. So again, thank you all for coming. I'm really jazzed to see so many people turn out on such a beautiful day. I hope you're all healthy and well and stay that way. So I'm going to share my screen now and launch my presentation. And as Denise mentioned, I'm going to speak for about 40 minutes. I plan to finish at about 3:45. And then we'll have 15 minutes for questions and answers. And I'm just gonna, you're welcome to submit your questions during the presentation, but I'm not going to address them until the end because my concern is I won't be able to get through all that I want to get through. And so this is a presentation. Well, I'm gonna kill my audio now.
Not your audio, but your camera.
My video, sorry. Yeah.
Very good.
Okay, so I just knocked off my video. So this is a presentation. It's very similar to the one that I gave at the Botanical Symposium in March. I'd like to say that this is better than that one, in part because I think I tried to cover too much material. So I'm actually going to cover the same material, but in slightly greater depth, and I hope more clearly. And hopefully you'll get a lot out of it. I feel like there's some interesting things here to share with you. And so I just wanted to point out that this is a picture taken by Gary Brill at Black Canyon. And Black Canyon is in the Venus area between Klickitat and Yakima County. If you get a chance, if our world changes, you can go out and do wildflower hikes. It's a great place to go in May and early June. Okay, so an overview of the talk. I'm going to first start off with some updates to the Flora Project website that I think are particularly interesting and exciting. It's going to require me to close this out and open up my web browser. Should go seamlessly. I'll talk about additions to the flora, notable taxonomic changes, and then some notable nomenclatural changes. So, just as a reminder, there is a website associated with the Flora second edition, and there's the URL here at the top. There's a bunch of categories that you can select for different pieces of information. Under the treatments category, you will find what I think might be the most interesting things to people. That is the errata in the book, slight errors, and then updates. We've started adding distributional changes and some other information. So And then we also have some revised treatments. And so what I'm going to do now is get out of here and switch to my web browser so that we can actually do this live so you can see. So here's, again, we're at treatments. If I go to errata and minor changes, you can see that this was last updated on March 20th. And then the changes just occur by page number. And so I'm actually, Ben Legler and I are actually working with an undergraduate student who we're gonna change the interface so that you can select whether you want a updates, nomenclatural changes, whatever. But anyway, currently all of those changes occur on the basis of the page, and you can see sometimes we have some suggested wording. As opposed to here, we have some additional distribution information. And these are just minor things that you could make a notation in your copy of the book. And I would say if any of you are using the book and you come across errors, please send them to me. As painful as it is to receive them, it's really helpful correct. And I should mention that I just found out from the UW Press that the second printing of the book has arrived in the warehouse this past week. And so many of the errata on this page were captured in that second printing, and the quality of the illustrations has been improved. So my understanding is those books are at the warehouse. I don't know exactly when the first print run runs out and they start sending out the new ones. And I haven't even had a chance to go to see the new print run because of our lockdown, and I don't feel, as curious as I am, it doesn't feel like an emergency, so I'm just going to have to wait to see it myself. But I have heard from the press it looks quite nice. Okay, so there's that. The other thing I wanted to mention is that we have started posting revised treatments, and I have some good news. The Washington State Department of Transportation accepted my $20,000 grant proposal today to hire Ben and Peter Zika, myself, and Crystal Shin to begin producing additional revised treatments, and that work will start May 1st. And so what we're going to do is start posting revised treatments because, as you'll see today, new information has become available. We want to include that information without having to print a book. And so you may recall that Berberidaceae has been changed slightly to recognize Mahonia and Berberis as distinct from one another. And so I have prepared a updated Berberidaceae treatment that has a key to those genera and has those species split out between those two genera. So we have 3 species of Berberis and 4 species of Mahonia, and you could download that and slip it in your book, or slip it somewhere else to keep on files and have that information available to you. So, we're going to start doing this more regularly over the coming months. So, I'm really jazzed to have that capacity and functionality set up such that we can do that. Okay, so that's all I had for the moment on the Flora project website. So let's get to my back to my presentation. Oops. Okay. On with the talk. So, additions to the flora. And I just want to mention that there are a couple of ways that things get added to the flora. We could have taxa new to science. For example, a new species, a new subspecies, a new variety. We could have existing native taxa that were not previously known from Washington, that have been discovered here in Washington through ongoing field work, or, as we'll see, review of historic specimens. And the same is true that there are non-native taxa that occur elsewhere that have never been documented here that we come across each year. And for several years, about 15 in fact, I have been managing the Washington Flora checklist. The website, the URL is down here at the bottom. And through the Washington Flora checklist, we track the name of every taxon, minimum rank taxon, that would be species, subspecies, or variety, that has been documented to occur here in Washington. And so if we just go back over the last 6 years, starting at 2014, you can see that there has been a steady increase in the number we have documented here in Washington. I do need to point out the sizable increase between 2017 and 2018, and that has to do with the fact that that's when the Flora second edition was published, and we incorporated all those taxonomic and nomenclatural changes into the checklist, such that there was a considerable number of changes. I'm seeing a lot of flashes here, and I feel like hopefully there's not a critical problem. Denise, are you there? Okay. Anyway.
Yeah, David, it's okay.
Yeah.
We are experiencing a little bit of bandwidth issues, especially when you went online, but it's much better now.
Okay.
Thank you.
Alrighty, so you'll notice though, between 2018 and 2019, we actually only added 9 new taxa, which is by far the smallest number of increases over the last 6 years. So it's not due to less field work. It could be that, we've found not all, but most of the things that are here. But as you'll see, we still have, there's good evidence that we haven't actually recorded everything. So let's start out with the first category, taxa new to science. And so in this paper that was published in 2019, Barbara Erder, who is really the world's authority on the genus Potentilla and Drymicalis, she published a new variety of Potentilla that occurs from Alaska down to northern Washington. And she calls that taxon Potentilla jepsonii, variety cluanensis. And there's a national park up in the Yukon, Kluane, I believe, and that's where the name comes from. And in her publication, she cites a specimen collected by Art Krugerberg from 19 Mount Chapaka as one of the specimens of variety cluanensis. And so here's that specimen right here. And this is a picture from the publication. Here's Potentilla jepsonii, variety cluanensis. Here's its closest relative, Potentilla pennsylvanica. And it's worth pointing out these two names here, because if you go to our book, you're not going to find the name Potentilla jepsonii. That's because the author, Peter Zika, decided that based on our specimens in the collections, he wasn't able to distinguish the species Jepsonii from Potentilla pennsylvanica. So what he did is that he synonymized Potentilla jepsonii within Potentilla pennsylvanica variety Ovium. So after discussing this with Peter over the winter, he has agreed to go revisit this treatment and see whether or not it is possible, upon further review of Barbara's paper and our specimens, and see whether or not he can separate out these two species, Jepsonii and pennsylvanica, such that we can accommodate this new variety. The reason why it's of note is because the Washington Natural Heritage Program is tracking this taxon, this variety of Potentilla jepsonii. In fact, in the course of field work last summer, Walt Fertig found some populations of it up around Mount Chapaka. So, you know, we want to have as harmonized a treatment of our flora as possible, one that, you know, harmonizes conservation interests and our ability to recognize things in the field. I mean, we need to have keys that field botanists can work, that have reliable distinctions, such that we aren't just guessing what things are. Okay, so that's pretty exciting. Gary Brill, who I mentioned, was the photographer of the photo on the first slide, has done a lot of backcountry travel, hiking, backpacking, climbing, and and he was in Fife's Peak, and this is a view from Mount Aix looking to the north towards Fife's Peak here. And if you're familiar with this area, where we are is just to the east of Mount Rainier. Mount Rainier would be off to the side here. Highway 410 would be running in this valley between these two ridges and going up to Chinook Pass. And so one thing you'll notice from this photo is the color of the soil in this area. And in conversation with Mark Darrick, he mentioned that that coloration is because these soils are derived from past volcanic activity. And they seem to support a kind of a interesting mix of species, which probably explains why a lot of rare plants occur on Mount Aix. But Gary was hiking around in the Fife's Peak area, and he came across this Lomatium. And Mark Darrick, as I mentioned, who is also has background as a geologist, he's an expert in Lomatium. Gary showed him this plant, and Mark this photos, and Mark actually did some field work. And Mark is involved with the group down in Boise that does molecular studies. So they took some tissue of this, they ran it through their DNA analyses, and it turns out that it's based on morphological and molecular evidence suggests that what Gary found is a new species of Lomatium. And it seems to be kind of restricted to that area between Fife's Peak and Mount Aix. So Mark and his colleagues have put this in the queue of Lomatium species that they need to work on and describe here over the next couple of years. And so they got a grant from the group down in Boise, got a grant from the National Science Foundation to pursue these things. So I think that we'll see some results from that work sooner rather than later. So that's pretty cool. So, you know, again, go to unusual places with unusual soils. One can encounter unusual plants. That's kind of a recurring story everywhere, and in Washington as well. So that brings us to our next category of existing native taxa newly documented in Washington. And so, strangely enough, over the past year, well, actually, before I get to the another one, there have been a couple of interesting finds. This is a species of plant that I was not familiar with, Micropus californicus. It's in the Asteraceae. It was photographed by Bob Carr. Bob is a retired botanist from Eastern Washington University, and he and his brother Jerry have contributed about 20 to 25,000 images in the Plants of Washington Image Gallery. They were down in Klickitat County, and they came across this growing along the roads. It looks like Gamakita, Studentophallium. It's in that group, and so they're actually pretty knowledgeable in that group, and particularly Jerry Carr. And they recognize this as Micropus californicus. And that's the first, well, that is the first wild growing population of Micropus in Washington. And the next closest populations occur in the Willamette Valley south of Portland down to California. So, you know, Willamette Valley is hot and dry. Klickitat County is hot and dry. you know, it seems like this is not a huge range extension, seems entirely believable. Interestingly enough, there is a specimen of Suxdorf's who, as you may or may not know, had a house here along the Columbia River in Bingen. He called Falcon Valley. And he lists that plant as growing in his garden there. I'm assuming he brought it in from somewhere. But the distance between Falcon Valley and this location where Bob Carr imaged it is well beyond what we would expect just from a casual escape. And so I really think that what Bob Carr found was a wild native population of the species. And that being said, nobody has gone down and gotten a specimen yet. So if you're down in that area and are descending from Goldendale down to Lisle or down around Packwood, take a look along the side of the road there. If you see this thing, please grab it, make a specimen, and send it to us. Okay, so strangely enough, over the last year, we added 2 native alpine dandelions to our flora. And one comes from a historic specimen with a strange story, and one comes from a contemporary specimen. So let's start with the old one. So this specimen was collected by Thompson in the 1930s at Mount Baker. And it was filed, you can't see, I should have done a call out of the label, but when Thompson collected this in 1935, he just called it Taraxacum. He wasn't sure which species it was. And he collected on Chowder Ridge at Mount Baker. And then in 1985, someone came and visited the collections, and they identified it as Taraxacum scopulorum. But they put the annotation label inside the fragment folder here, and nobody knew this was in here. And then the specimen got accidentally misfiled in the Taraxacum lyrata folder, such that this got overlooked in the course of writing the flora. And then sometime in the last year or so, I was going through the folders and I came across this specimen. I looked inside this fragment folder and it said Taraxacum scopulorum. And lo and behold, I've verified the identification using the Flora of North America treatment. And so Taraxacum scopulorum, discovered, if you will, nearly 85 years after it was first collected. And this green dot here shows you where that locality is. And as you can see, it's the southernmost population west of the Cascades, and it's the westernmost population of this species here in the Northwest. It's much more common in the Rocky Mountains, and if you were to go further in the Rockies, Colorado, Utah, there's a ton of it. But this certainly is an outlier population, and something I'm sure that the Natural Heritage Program will begin to track. And, you know, we're particularly interested in these species at the southern ends of their distribution in the age of climate change. So certainly something we want to watch. Map below is used to work for King County Noxious Weed Program. He's an excellent field. Spends time with Cascades. He's turned up some interesting things. And last summer, he found a Taraxacum alaskanum just to the south of Cascade Pass. And so this is a picture of it here. He brought this specimen in, so we have a specimen of it. He and I went over the identification of it using the Flora of North America key. And so we are quite confident that's what it is. This shows you the distribution of Taraxacum alaskanum in North America. And this is the population down here. So again, another situation where we have really what is the southernmost. It's not the westernmost population because we have some populations out here on Vancouver Island, but definitely the southernmost Taraxacum alaskanum population. And in addition to our flora, and again, something else that's worth keeping track of, monitoring the potential impacts to this is an interesting to the floor that I'm going to take a few minutes to explain some of the biology behind this particular find. But again, Thompson, the person who found the Taraxacum up on Chowder Ridge in 1935, he collected a Bougueria, which was split out from Arabis, on Table Mountain in 1935. The guy got around a lot. And so it turns out that it was a new species. It was described in 2007 as part of the Flora North America treatment for Bougueria as Bougueria cascadensis, and it was only known from that one collection. That's the only specimen of it that has ever been documented. So we call that's a type specimen, and that's the type locality. In 2013, I was collecting in Yakima County, and I grabbed a Bougueria. And because while I was working on the Flora project, I didn't get around to identifying that specimen until after the book was published. And it turns out that this too is Bougueria cascadensis, which made it the second known collection at that time of this species. And it's an interesting species because it is a triploid apomictic hybrid of Bougueria microphylla and Bougueria pepercola. So I'm going to explain what triploid and apomictic mean, and I will also show you the range of these two species to show you why this is kind of an interesting discovery. And then some additional information that has come available since I gave this presentation just a month ago. Okay, so the two parents, Bougueria microphylla, Bougueria pepercola. Bougueria microphylla is widespread in the East Cascades in Washington. So this is the Cascades here. You can see it's all in Yakima and Klickitat County. No surprise that it would be the parent of hybrid offspring. Bougueria pepercola is a stranger story. There are two historic populations of it from the Olympic peninsulas, like the 1930s is when I say historic, and no other collections in Washington. So how did a hybrid between these two species form in this general area? I don't have an answer to that. It could be that there's more Bougueria pepercola in that area than we know. Bougueria is a really hard genus of plants to identify, and so certainly be worth maybe going and taking a look and see if there's more of it there. But anyway, that's part of the interesting story. Let's talk about triploid. So triploid means it has 3 copies of a chromosome, 3 copies of each chromosome. And N refers to the ploidy level. So we get triploids when we have a hybrid between a diploid. Humans are diploid. You get 1 set of chromosomes from your mother, 1 set of chromosomes from your parents. We're 2N. Plants have the ability to duplicate their chromosomes without problems. So you get tetraploids, octoploids, dodecaploids, all these crazy things. So in this case, a diploid, one of the parents is diploid, one of the parents is tetraploid. They hybridize and they form tetraploid offspring. So what does that mean? Triploid offspring, those triploids can't backcross with either of the parents. So you don't get this hybrid swarm. Some of you may be familiar with the way that balsam roots hybridize when they come together, and you get all these crazy intermediates. You're not going to get any crazy intermediates here. These things they only can reproduce with themselves if they can reproduce at all. But this is a particularly different set of plants because they are apomictic. And apomixis is a form of reproduction in plants where you get fertilization, you get reproduction without fertilization. That is, there's no fusion of gametes. You get this spontaneous production of seeds. Dandelions are our most common and abundant example of apomixis around us. We can think of it as a form of asexual reproduction because there's no gene exchange, there's no pollination, there's no, these things are just forming these seeds through these crazy interactions in their cells. And so they're on their own evolutionary trajectory that is distinct from either of the parents. So that's why we assign a name to this hybrid, and that's why this hybrid is of interest. Those hybrid swarms that you see among the balsam roots, they're interesting, they're cool to look at, but they're never going to form a distinct species because of so much intermixing. So that's Buchera cascadensis. So since I gave this presentation, we added some specimen data from the Royal British Columbia Museum in Victoria. And it turns out that Curtis Bjork, some of you might know Curtis, he's an excellent botanist. He used to be at Washington State. He lives up in BC now. He went through the Buchera collection at RBCM and found several from the Manning Park area and nearby. So I found the second one ever. Not true. There's about 4 or 5 populations in this area. So that's an exciting discovery. And this is the kind of thing that we will include in our updates to the website, and why we need to continue to update the information on that website, because new information is constantly becoming available. Okay, so those are the new taxa added to our flora. Let's talk about a couple of non-native ones. One is particularly hard to overlook, and that is a non-native member of the genus in Washington, even here west of the Cascade Crest, is up in San Juan, Whidbey Island. Though our Opuntia have roundish pads, roundish stems. They kind of look like the length and size of your thumb and thickness of your thumb. This is one of the flat-padded Opuntias, and this is a distribution map of this species from the Flora, North America. You can see it's native in the southwestern US. Peter Dunwiddie and Peter Zika collected this in Klickitat County this past year, going along some railroad tracks. There's about 40 or 50 plants growing there. I have no idea how it got there. It's possible some cactus enthusiast thought, well, I wonder if this stuff will grow here. So anyway, it's flowering there. And let's hope that it doesn't spread too much further than that. And maybe someone will go down there and eradicate it. I don't know. The other notable non-native addition to our flora is a species of Polypogon. We have a non-native Polypogon that's real common. It's called Polypogon monspeliensis. Well, this is quite different. Again, Peter Zika collected this down in Klickitat and Benton Counties. And to give you an idea of what a range extension it is, this is the distribution of Polypogon maritimus in California, really widespread. It goes up to southern Oregon, all south of Utah. So populations along the Yakima River are a sizable range extension. And I'm guessing that there's more of it around than just those 2 populations, because the differences between the 2 species are quite subtle. So these are illustrations from the Flora, North America grass volumes. Here's our common Polypogon monspeliensis. You'll see this up in the San Juans, both sides of the Cascades. It's kind of neat looking annual grass. But this is the spikelet. These are glooms, and they have these real long awns, and they've got a single floret in there. And you can see the floret in monspeliensis has a long awn, but in maritimus, the glooms here have these long ciliate hairs, and the floret lacks an awn here. So if you're a grass enthusiast, out looking around, you see Polypogon, it's worth taking a look, see if maybe you have this as well. Okay, so notable taxonomic changes. Coming down the home stretch here. My clock shows 3:37, so I'm going to try to wrap this up. And the nomenclatural changes in the next 8 or so minutes. So notable taxonomic changes. Some of you might be familiar with Syntheris. They're really beautiful members of what used to be the Scrophulariaceae. Now they're in the Plantagenaceae. There's some up in the Olympic Mountains, more common in the Columbia River Gorge and east of the Cascade Crest. They now belong to the genus Veronica. And why is that? Because molecular evidence has shown that it is closely related to Veronica. And I realize this is a really busy slide here. There's just a couple of things I want to point out. First is that this study was now 10 years later. It just shows you how long it can take for information to make its way into local printed floras. The other thing I want to point out here is here's Syntheris. This is just a tree of life, if you will, that for a certain portion of the tree. And everything from right here upwards is considered Veronica. And so you'll notice that Syntheris is this clade, this group that's smack dab in all of these related lineages. And we know they're related because they have very strong, what we call bootstrap values or other indicators of the confidence we have in the data, the strength of the relationship. So these little asterisks that you see throughout the tree here represent 100% certainty. And then these smaller numbers at these different nodes are less than 100, but they're mostly between 85 and 98, which means this phylogenetic backbone, this tree is very robust, and these results are not going to be changed by further studies. And so when we have groups squarely planted in the middle of a larger group, then we have to call it by the name of that larger group, which in this case is Veronica. Some other things that got caught up in this study are the Hebes. Obviously, they're not native here, but if you're west of the Cascade Crest, you might be growing them in your yard. They are now Veronicas also. So that's going to require us to redo the Veronica key and put all of those Syntheris into the Veronica key. So that's the kind of thing that will get covered in this new round of funding. A Veronica or Syntheris that you might have been familiar with, which is now Veronica, is our endemic Syntheris lanuginosa, which is endemic to the Olympic Peninsula. It's now Veronica dissecta, variety lanuginosa. Okay, so I just want to quickly go over what we hope to make a change. Most of you are probably like me, and that you learned that the maples, the genus Acer, belong to the family Asteraceae. And so there has been some molecular work that was done that is going to result in it. So recently, over the last 20 years or so, Acer was placed in the in the family Sapindaceae, which is mostly an Asian and tropical family. But the molecular evidence and the rules of doing molecular studies made it such that everything that the Acers had to be put in Sapindaceae. Now, what they did is they created a small family here that allows all of these segregate groups to be treated as their own families. Included in that is Asteraceae and Hippocastanaceae, the chestnuts, and molecular evidence shows that Asteraceae is a distinct group of plants from Hippocastanaceae. And you would agree Asteraceae is a morphologically well-aligned group. They all have opposite leaves. They produce the samaras. So we are likely going to, in the next iteration of revising the book, treat maples back as Asteraceae. Okay, the last thing I want to cover is notable nomenclatural changes. At the family level, you may have noticed in the Flora second edition that we treat the family Adoxaceae as separate from the Caprifoliaceae. And Adoxaceae in our flora for the Pacific Northwest includes Adoxa, sambucus, and viburnum. Adoxa occurs in Montana, so in Washington we only have sambucus and viburnum in Adoxaceae. Well, it turns out that Adoxaceae was published after the name Vibernaceae. And so the rules of nomenclature say that you have to use the earliest published name when naming a species, a genus, or a family. So an effort was made to conserve the name Taxaceae. That failed. So going forward, we have the Vibernaceae that will include Sambucus viburnum, and if you're in Idaho, it'll also include Taxa. So the last thing I want to cover is something that I mentioned 2 years ago. What do we do about Rubus parviflorus? Because Rubus parviflorus technically is what we call an illegitimate name. And what do I mean by that? Illegitimate names are typically ones that were published after somebody else published that name previously. So in the original Hitchcock, and even in Flora, North America, you have Rubus parviflorus. The author is Nuttall. It's abbreviated as Nut. Turns out I'm going to show you that that's an illegitimate name. And so what the next earliest name used by botanists for this plant that we call thimbleberry was Rubus nutkanus. And those are the authors, and that's what we used in the Flora 2nd edition. And so I don't make the rules, I just have to follow them. It turns out that Rubus parviflorus was published by a gentleman in Europe in 1770. Obviously, people didn't have access to the literature. People in North America didn't have access to European literature in 1818 when Nuttall published Rubus parviflorus. So since this is a later name, this name becomes illegitimate. If you go to the International Plant Names Index, they show Rubus parviflorus as illegitimate. Fortunately, there is a recourse in that you can write a paper and submit it to the Cultural Committee for consideration, that you can do what's called conserve a name. And so that is what the Flora of North America author for Rubus has done for Rubus parviflorus. He submitted a publication saying, listen, this name is used like everywhere. It's widely distributed. It's used in nearly every flora. Let's keep the name Rubus parviflorus. And I somehow got dragged into this debate because it's in the course of that publication, I've just cut and paste out of it. He says he agrees that Rubus nutkanus is the next available name. It says, so far, Rubus nutkanus has been taken up as the accepted name for this species only by Giblin et al. Well, technically, the citation is Hitchcock and all, but also, this is the only flora that has been published. So it's not like we are acting rogue. We're just following the rules. So the take-home message here is that remains to be seen. In many ways, I hope we do retain the name Rubus parviflorus because it is what people know, even though it's not a good descriptor for that species because it's one of the largest flowering rubus that I'm aware of. All right, so that is all I had. It's 3:45. I did want to acknowledge some of the people who made today's presentation possible. Cody Hinchliff and Joey Ellis and Ben Legler all gave me a lot of help in putting together that flora, the phylogeny that I showed you. Oh, I didn't show it. I have to. Oh, let me see. I want to just go back here quickly. Nah. You said it. I think that this is what I wanted to show. We produced a phylogeny of all the families in the flora. And this is really cool because it shows you the total number of non-native taxa, of families represented by non-native taxa. If you scroll over a family, you can get a list of all the genera in that family. Go down there to the bottom, larger clades. I don't have a lot of bandwidth here. I'm going to stop there, but I would encourage you to explore that part of it here. Again, it just shows you the non-native families, shows you the evolutionary relationships among those families, and we're really jazzed to have that up. Okay, so I will stop that, and I will go back to my presentation and finish with my acknowledgements and then answer any questions that people have. So Cody, Joey, and Ben gave me help with that phylogeny. The Nygrum Family Endowment for the Herbarium and Friends of the Herbarium support the field work that I do. And I will stop screen share and I will go to question and answers. Okay, so a question came up. Where do you draw the line on taxa and where they are found? If someone brings a new plant in and puts it in their garden, what about horticultural varieties, at least the heirloom types? Okay, so we definitely make a distinction between cultivated and non-cultivated taxa. And so sometimes cultivated taxa or species escape into the wild, and when they naturalize, then they reproduce, they establish on their own, they maintain themselves without cultivation, then we would consider them part of the flora. But if somebody, I got an email from a colleague who lives up in the Okanagan. She said, "Hey, I found this Trillium species, Trillium albidum. It's growing in a land parcel out here. There used to be a fish hatchery. That species only occurs west of the Cascade crest. Do you think that this is wild?" And the answer is no. That has just persisted from cultivation, and we're not going to consider that a wild plant. So that's
David,
my answer.
There's another question also in the chat.
Okay.
And participants, this is a good time to ask your questions by using either the Q&A or the chat.
Yes, he's done some. Bucher, would the amount of genetic diversification that has accrued be a measure of time since its formation? So just to read the question, regarding Bucher, would the amount of genetic diversification that has accrued be a measure of time since its formation? That is a great question that is really beyond my knowledge in terms of being able to answer in a specific way, but I can say this. Different lineages acquire genetic diversification at different rates. Some lineages just never diversify over long periods of time. And then other lineages like Castanea diversified rapidly over very short periods of time. So, how that translates into when species form, you're going to have to ask my boss, Dick Olmsted, or some other folks who study those kinds of questions. But great question. There's a question here. Out of curiosity, how many of the Flora did you print the first time? The first print run was 5,000 copies, and so was the second print run. So my understanding is that the 5,000 copies either was going to be run out in February or close to it. So yeah, there are now 10,000 copies in print.
And Ellen Watros has her hand raised. So Ellen, I'm going to unmute you to ask your question. Ellen, are you there? Oh, maybe not. Okay, more questions.
I think that's it. Well, thank you. Thank you all.
Let's see. Thank you, David, for your presentation this afternoon. We did record, folks. Here's new questions. Here we go.
Yeah. Okay. Are there plans for a digital version of the Flora? There is an ebook version of the Flora that is available for Android devices. I'm not aware of it being available for iPads. Some of you may be familiar with the Jepson Flora that puts their Flora online. And the UW Press has not been supportive of that idea. They will allow us to put 20% of the flora online. And so we're thinking that when we get to that 20%, then maybe it's time to do a new book. And as to whether or not that's a printed version, I can't say. That's at least 5 years in the future, and what books look like 5 years from now is anybody's guess. Here's a great question. What is the difference between a variety and a subspecies? And if you talk to people who study animals, they have a very different idea about the difference between varieties and subspecies. At one time, I think in plants, botanists differed in their view of variety and subspecies, but over time, whatever the definitions were have kind of merged into one another. And for all practical purposes, there is not a difference between a variety and subspecies. So one could say, well, what's the difference between variety, subspecies, and species? We assign a variety or subspecies to a portion of a species that shows variation in a predictable way, whether that's geographic or environmental. And by environmental, I mean by soil type, soil moisture, elevation, those kinds of things. So some environmental or geographic difference in some detectable morphological character. It might be hairs on the leaves, it might be petal color, it might be the size of the fruits or something. Some variation that's predictable gets assigned a variety. Typically when you encounter two forms side by side, we just say, well, it's just a morph and it doesn't warrant, when I say morph, it's just a form and it doesn't warrant taxonomic distinction.
Okay, well, we have a thank you, David. We have wealth of info, thanks, applause, thank yous.
Yeah, so thank you. Well, I just wanted to say thank you all, and feel free to email me with any follow-up questions, or if you'd like me to post the presentation, I can do that. Gladly share it with anybody who's interested. And thank you, Denise, for hosting me and inviting me to share today.
Well, thank you for being part of our Native Plant Appreciation Month webinar series. David will be offering a second webinar next week. We have changed the time from 4 to 4:30 on April 15th, and it will be a hands-on tutorial using the Consortium of Pacific Northwest Herbaria online database. So we hope you'll all look for that and watch for announcements on the posting of these videos that we're recording during this webinar series. Thank you all, and enjoy this beautiful afternoon. Thank you, David.
Thank you. Bye-bye.